Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, let's unpack this. Imagine a surgeon, right? They're in the operating room, total focus, the tension's high, and they're working meticulously to remove a tumor. It's just one of the most critical moments for a patient, and the surgeon's success. It often really hinges on this one crucial but incredibly difficult thing, knowing exactly where the tumor ends and where the healthy tissue begins. It's a line that can be surprisingly blurry, and, well, getting it wrong can unfortunately mean more surgeries for the patient or even, long-term damage. But what if there was a way to give surgeons, like, a shortcut to much greater precision? A breakthrough that honestly sounds a bit like science fiction. It involves something called a phantom. Now, we're not talking ghosts or anything spooky here, no. This is a meticulously crafted, three-dimensional printed model, and it's designed to perfectly mimic human tumors. It's actually the opposite of scary. For patients needing tumor extraction, this could be a huge step forward, a real advance in a promising surgical procedure.
Yeah, and what's truly fascinating, I think, is how this innovation directly tackles a really long-standing, very tricky challenge in cancer surgery. So our deep dive today, we're going to peel back the layers on this groundbreaking technology. We'll be drawing insights from a recent health news piece and also a really detailed scientific paper in ACS Nano. And we'll explore how these incredibly realistic models could genuinely revolutionize how we approach removing tumors.
Okay, so let's start right there. From the surgeon's point of view, what makes removing a tumor so challenging? I mean, on the surface, it seems like it should be straightforward, just, cut out the bad stuff. But clearly it's not.
You've really hit the core issue. The main goal, the ideal outcome, is achieving what surgeons call negative surgical margins. Basically, that means after the tumor's out, there's absolutely zero cancerous tissue left behind right at the edge of where they cut. And that's absolutely critical for minimizing the risk of the tumor growing back. But here's the kicker. Tumor margins are often really indistinct, irregular even. Because cancer, it tends to grow invasively, right? Sending out these tiny microscopic tendrils. So this often leads surgeons to, remove a larger area of healthy tissue around the tumor just to be absolutely certain they got it all, which is understandable, but depending on where the tumor is, that can unfortunately cause things like neurological damage or just generally reduce a patient's quality of life. In the operating room itself, surgeons mainly rely on, their sense of touch. That's called palpation, and just looking very closely, visual inspection. Trying to feel and see the difference between the tumor and the healthy tissue. But relying on just those senses can be risky. Cancers often aren't easily felt, they're not palpable, or they don't look obviously different. And that leads to surprisingly high rates of what are called positive margins during surgery, which means some cancer cells were actually left behind.
Right. That immediately tells you we need something more precise than just sight and touch.
Yeah.
And this is where the advanced imaging comes in, like fluorescence image-guided surgery. It sounds amazing, offering real-time feedback during the operation. But you're saying even that has its own limitations. It's not quite the perfect solution yet?
You're absolutely right. Fluorescence image-guided surgery is it's a really promising technique, a big step forward, but it definitely faces some significant hurdles. A major one is what scientists call the limited penetration of photons in biological tissues. Basically, light just doesn't travel very deep into our bodies. Think of trying to shine a flashlight through, thick mud. The light gets scattered and absorbed quickly. And on top of that, sometimes healthy tissues themselves can emit a faint glow. It's a natural phenomenon called autofluorescence. This creates a kind of background noise or static, making it harder to clearly see the specific glow coming from the tumor that you've tagged with a dye.
Okay, so it's like trying to see a faint star next to a bright moon.
Exactly, that's a good analogy. And the current dyes that are approved for clinical use by the U.S. Food and Drug Administration, things like indocyanine green and methylene blue, they typically operate in a specific light range. It's called the near-infrared one window, which spans about 650 to 900 nanometers on the light spectrum. Now, these dyes have their uses, but they also have drawbacks. One is photobleaching. Imagine a bright color fading quickly in the sun. The dye basically degrades under the intense lights used in surgery. They also have limited brightness to begin with, and crucially, poor tissue penetration, often only about 3 millimeters deep.
3 millimeters, that's really not much at all.
No, it's often not deep enough for many types of tumors.
That really does highlight the challenge. And what about how we test these new imaging agents, these dyes, before they even get close to being used in people?
Yeah.
I understand there's a pretty fundamental issue with relying so much on animal models, like mice, in the early research stages.
Yeah, that's a huge point, and it raises a really important question for the whole field.
Mhm.
How do we accurately test these new agents when the animal models we use just don't truly replicate human anatomy? Assistant Professor Indrajit Srivastava, who's leading some of this new work, he explains it very clearly. He says, mice skin is very thin, only a few millimeters, whereas for humans it's very different because we have layers of fats, muscles, and tissues that can actually affect the performance of the imaging agent.
Right. Makes sense.
So he points out mice are actually not the best model for these studies because of this basic difference in tissue depth and complexity. And this discrepancy, this gap between mouse models and human reality, it means that many promising new imaging agents, things that look great in a mouse, they unfortunately never make it to the operating room. They just don't perform as well when tested on larger subjects or humans. It's a real bottleneck stopping progress.
Okay, so we've got this picture. Surgeons facing these blurry lines, current imaging hitting depth limits, and the testing process itself being hampered by, well, mice not being tiny humans. But this is where it gets really interesting. This is where the innovation from Texas Tech University's Edward E. Whitaker Jr. College of Engineering comes in. Spearheaded by Assistant Professor Indrajit Srivastava and his team, including folks like mechanical engineering doctoral student Asma Haroon and former biochemistry undergrad Nate Bendelin, they've developed these, frankly, extraordinary three-dimensional printed tumor-mimicking phantom models.
And they really are extraordinary. These aren't just simple shapes. These phantoms are meticulously designed to replicate not just the physical form, but also the optical properties of human tumors, how they interact with light. They're usually a few centimeters long, and they're built using a careful mix of substances. Things like lipids to simulate fatty tissues, hemoglobin to mimic blood content and how oxygenated the tissue is, enzymes for biological activity, even actual tumor cells sometimes, and it's all bound together with gelatin, which gives it a firm but realistic structure. Then they're three-dimensionally bioprinted into various shapes that actually mirror real tumor forms, segmental, spherical, discoidal, reflecting the actual shapes of cancers like breast cancer tumors. It's really an amazing blend of engineering and biology.
Wow, that level of realism is incredible. So what does this actually mean for research, for developing those better imaging agents we need?
Well, it means these models can potentially eliminate this need to go back to the drawing board so often. When an agent looks good in mice but then fails in later tests, these phantoms offer a much more realistic intermediate step. As Professor Srivastava puts it, if a nanoparticle or an imaging agent goes through the artificial tumor, it can actually mimic that environment and its optical signals will be affected like how it would be in human patients.
Ah, I see.
So researchers get much more accurate feedback much earlier on. And not only that, he adds, this is actually a much cheaper alternative than animal studies because you can do it a lot more to get statistical significance.
It's cheaper and faster.
Exactly. It can dramatically speed up the whole research cycle and makes testing more efficient, more accessible.
That's incredibly powerful. Faster, cheaper, and more accurate testing.
Mm-hmm.
But how do they actually make them behave so much like real human tissue? What's the kind of secret sauce for that realism?
Yeah, the choice of materials is absolutely key. For the main structure, the sort of scaffolding, what they call the bulk matrix, they found gelatin worked much better than something like agarose.
Right.
Gelatin could maintain these firm and rigid structures and, importantly, had no observable leakage for up to 3 days, even at different temperatures, which is important for practical use. Then, to replicate how light interacts with tumor tissue, how it scatters and gets absorbed, they use specific ingredients. They use intralipid as the scattering agent. That simulates the density and the sort of unevenness, the heterogeneity of tissue. And they use hemoglobin as the absorbing agent to model the different levels of blood supply or oxygenation you find in tumors, which are often quite different from healthy tissue.
And what about testing the actual imaging agents inside these phantoms? How do the models help evaluate those specific tools surgeons might use?
Right, so the phantoms are designed so you can embed specific nanoparticles right into them for imaging, like these things called semiconducting polymer nanoparticles. Or they can even use actual tumor cells that have been pre-labeled with these nanoparticles at concentrations you'd find in a living organism. And what's really striking here is that these particular semiconducting polymer nanoparticles showed much superior photostability compared to that standard dye in the cyanine green.
The fading problem.
Exactly. Remember the fading, the photo bleaching? These new nanoparticles kept their signal strong for up to 4 hours.
Which is great for surgery times.
Precisely. That aligns really well with typical surgical timelines. And what's more, the tumor-mimicking phantoms themselves were optically stable for up to 2 weeks, meaning you can run longer experiments on them. And this also lets them dig into another important question. How well do these models capture the dynamic environment inside a tumor, the chemistry? They've actually managed to integrate key factors of what's called the tumor microenvironment. For instance, tumors are often more acidic than a healthy tissue. So when they exposed phantoms containing these semiconducting polymer nanoparticles to an acidic pH, the phantoms actually showed amplified fluorescence. They glowed brighter.
That's clear.
It is. And even more cleverly, they showed a signal-off response when they encountered glutathione, which is a molecule that's often found at higher levels in cancerous tissue, so it's like a smart switch. The signal turns off specifically where there's high glutathione, offering a potential tumor-selective mechanism, basically drawing a dark, clear line around the tumor against the healthy tissue.
Okay, this level of detail, this realism, it sounds incredibly valuable for developing better tools. But how does all this, fancy science translate into actually helping surgeons do a better job in the operating room?
Right. This is where we see the potential real-world impact. First, they did what's called quantitative imaging. That means they could precisely measure the relationship between how many nanoparticles were present and how bright the fluorescent signal was. And they showed a strong linear correlation, more nanoparticles, brighter signal, in a predictable way.
Okay, predictable is good.
Very good. Because this precise measurement ability is crucial for advancing applications such as multiplexed biomarker detection. Think of it like giving surgeons the ability to see not just one thing, but multiple different cancer-related signals or markers at the same time during the surgery.
Like different colors meaning different things.
Exactly. Potentially identifying different types of cancer cells or specific biological processes happening right there in real time. They actually showed the tumor-mimicking phantoms can be used to test this multiplexing optical imaging potential using 2 or more near-infrared nanoparticles, and they could effectively tell their signals apart.
So getting multiple pieces of vital info at once, that's a huge potential advantage. But what about that original problem you mentioned, seeing deep enough into the tissue?
Yeah, the penetration depth. To tackle that directly, they conducted what are called ex vivo optical tissue permeation profiling assessments. Sounds complex, but basically they took their tumor-mimicking phantoms and placed them underneath layers of pig tissue, porcine fat, muscle, and skin, because pig tissue is actually remarkably similar to human layers.
Okay, good stand-in.
A very good stand-in. And what they found was fascinating. Phantoms using nanoparticles designed for the near-infrared 2 window, a different light frequency range, showed significantly better performances than the standard near-infrared 1 counterparts. They maintained strong, clear signals even when viewed through an impressive 10 millimeters of fat tissue and 4 millimeters of skin.
Wow, that's much better than 3 millimeters.
Much better. And it's thanks to their deeper tissue penetration and also, very importantly, much lower background autofluorescence signals from the healthy tissues overlaying them. It's like having that much stronger flashlight that cuts through the fog better.
And bringing it all together, the ultimate goal, precise tumor removal. Did these models help show how a surgeon could achieve that clarity?
Absolutely. A critical feature for any imaging probe, any dye or nanoparticle, is its ability to accurately delineate the complete extent of the tumor margins to show exactly where the edge is. So, using these tumor-mimicking phantoms embedded inside healthy ex vivo pig lung tissue, they clearly demonstrated that certain nanoparticles provide much clearer signals with significantly lower autofluorescence from the surrounding healthy tissue. This essentially creates a dark background for healthy tissues, which is the ideal scenario for bioimaging, and especially for achieving that precise tumor margin delineation. It gives the surgeon an incredibly clear boundary to follow.
And they even simulated actual surgery, didn't they, to see how this would work in a more realistic, dynamic setting?
That's exactly right. They conducted surgery simulations on ex vivo porcine lungs. Again, pig lungs closely resemble human lung tissue. They implanted these tumor cell mimicking phantoms in different spots to simulate various scenarios you might encounter in thoracic surgery, like operating on lung cancer. And the images they got during these simulations, they were so clear, so effective, that they allowed for this successful resection, the successful removal of the phantom tumors. It really highlights the immense potential these models have to enhance surgical precision and outcomes when translated into real-world procedures.
It really does sound like this research marks a significant shift towards more human-centric studies. Moving beyond some of the inherent limitations of relying so heavily on animal models, it feels like maybe a turning point.
Precisely. And this work aligns perfectly with the direction major health organizations are heading. Both the U.S. Food and Drug Administration and the National Institutes of Health are actively encouraging this move toward more human-centric studies. Professor Srivastava himself addresses this head-on. He says, I think people have overused mice studies, at least from the perspective of showcasing their nanoprobes for surgery. But at the end of the day, the main priority should be how close the work is to actual clinical use. And to me, to the point, I think in cases like surgical navigation, my studies are more human-centric because we're trying to address the optical challenges faced by these imaging agents. We're actually accounting for how this technology would behave when you're using it in an operating room. And to back this up, they even performed a thorough comparative analysis. It showed that these tumor-mimicking phantoms can reliably replicate the optical properties of tumors studied both in vivo in living organisms and ex vivo outside living organisms. That's a really powerful validation of how realistic these models are.
So this deep dive really reveals how these innovative tumor-mimicking phantoms offer a truly novel and potentially powerful platform. A way to evaluate imaging agents under incredibly realistic surgical conditions. It seems like an approach that promises to seriously accelerate the selection of the best, highest-performing nanoparticles, bringing us much closer to a future where surgical precision isn't just incrementally better, but maybe vastly improved.
Absolutely. And the work isn't finished. Looking ahead, the team plans to continue refining these models. For instance, they're looking for better binding agents, things that can handle temperature changes during shipping better. That's a practical hurdle for getting these used more widely. And they're also working to keep the actual tumor cells alive for longer within the phantom models, maybe over several weeks.
Oh, interesting. Why?
Well, that could provide even more dynamic optical information, reflecting how a live tumor might change over time, which could be incredibly useful for a surgeon during imaging.
So thinking about the big picture, what does this all mean for you, for patients, for healthcare? Just imagine a future where every single tumor removal surgery, no matter how tricky, is guided by this kind of incredibly precise light, ensuring surgeons can remove all the cancer with confidence, leaving behind only healthy tissue. Think about what that could mean for thousands, maybe millions, of lives transformed. All thanks, potentially, to a meticulously designed phantom that, far from being scary, actually represents a real beacon of hope.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.